Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the next lecture of second unit of plant biotechnology course. In this unit we are learning about the ploidy. In the earlier lectures we discussed about the in vitro haploid production through androgenesis or gynogenesis. Then we also discussed about the polyploidy or the various methods of origin of polyploids, and in today's lecture we will continue talking about the polyploidy and we will learn specifically more about the triploids. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University, New Delhi. In JNU, I teach plant biotechnology and genetic engineering at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh, and this project has been sponsored by DTH Swayam Prabha, MHRD, New Delhi. So in this lecture, I will talk about the triploids, various methods used for the triploid production. Specifically, we will discuss endosperm culture in more detail and the various factors that impact the endosperm culture. And finally, towards the end of lecture, we will talk about the application of triploid plants in agriculture, horticulture or forestry or other associated industrial applications. Let's have a quick recap of the polyploids that we learned in our last lecture. So the first term was the monoploids. That is the condition when there is only one set of the chromosomes in their cells. It is called as the monoploid number or monoploid set of chromosomes. It is represented by the letter X. The next is the haploids. Now, haploidy represents the number of chromosomes in the gametes of an individual, and letter N is used to represent the haploid number. It is usually the half the number of chromosomes that is present in the somatic cells of that individual. The plants that have the haploid number of chromosomes in their body cells are called as the haploid plants. Then next term is the dihaploids. Now this term is associated with the gametes, and the gametes having two sets of the chromosome are called as the dihaploid gametes. And usually the gametes formed by the tetraploid individuals would have two basic sets of the chromosomes, and so the gametes of the tetraploid would have a dihaploid ploidy level, and they will be called as dihaploids. Next is a diploid. So the plant have two sets of the chromosomes in their body cells, and a pair of same chromosomes from two sets, they are same or identical, and they are called as the homologous chromosomes. So basically the plants having two sets of chromosomes in their body cells, they are called as diploids. And then we talked about polyploids. So basically if the body cells of the plant have more than two sets of the chromosomes, this condition is called as a polyploid condition or polyploidy, and the plants are called as a polyploid plants. And depending upon how many chromosome sets are there in the cells, these are named as triploid for if there are three sets. If there are four sets of chromosomes, these are called as tetraploids, and if there are six sets of chromosomes, then they are called as hexaploid. And similarly, other terms, octaploids, decaploids, can be used depending upon the how many chromosome sets are there. Now, polyploids may be autoploids. It's another term associated with the polyploids. So this is a form of a polyploidy where all the chromosome sets of the body cells, they are identical or they are same, or basically they originate from the same species, like this AAAA. It means letter A represent the one chromosome set, and it says that there are four set of chromosomes. However, they are replicated from the same chromosome set, and so they just originated from the doubling of the chromosomes from particular species, and they are called as autoploids. Second term is alloploids or allopolyploids. Now it's another form of the polyploidy where the chromosomes present in the cells are contributed by more than one species, like AABB. So here A and letter B, they represent the chromosome sets, and different letters are used here to represent that A belongs to one particular species A and B belongs to another species. So the chromosome sets here are contributed by two different species, and that's why these are called as the allopolyploids. Then we also talked about various mechanisms of origin of polyploids, where we discussed about the production of unreduced gametes because of the meiotic errors that usually result due to the environmental stress conditions. Then we also talked about polyspermy, that is the fusion of more than one male gametes with the egg nuclei, and also the chromosomal doubling either in the zygote or in the somatic cell can lead to origin of the polyploids. And finally, we talked about various applications of the polyploids, where we discussed about the polyploid as a major driving force for the evolution of the angiosperms or the land plants. We also talked about their roles in the crop improvement or induction of stress tolerance, and their application for the industrial purposes. So today we will talk about or we will learn more about the triploids. Now, triploids means that each cell has three sets of chromosomes, that is, three copies of each basic chromosome set are present in these in the cells, in the each cell of these triploid plants. Or in other words, it can also be explained as that each gene has three allele in these individuals. So here is a pictorial representation of a triploid individual where each chromosome has three copies, and in total there are 5 chromosomes. So in other words, triploids have 3 sets of chromosomes, and each set here in this condition, this pictorial representation, has 5 chromosomes. So that means X is equal to 5 in this case, and so we can also say that the monoploid chromosome number in this situation is 5. Triploids have several desirable characteristics, such as they usually have larger organs, like their fruits will be larger, they have big flowers, or their leaves may be larger or thicker. Leaves are usually broader or thicker. They are dark in color and or dark green in color that result in a great vigor and also increases the efficiency of the biomass production. Usually these plants have a better stress tolerance capabilities too, so they perform better in the environmentally unfavorable conditions or when the limited amount of resources are available. Triploids are sterile plants and so they do not bear seeds. Therefore, they bear seedless fruits and which are specifically desirable for the fruit industry. Several triploid fruits are available in the market that we use on the daily basis, like seedless grapes or the seedless watermelon. Similarly, the seedless citrus fruits like the mandarins. So overall, these plants have or the triploids have a greater economic values and has huge impact in the agriculture, horticulture or forestry. Next is the methods of the triploid production. So there are actually three major categories where the triploid production methods can be grouped. So one is the natural selection, then we have artificial hybridizations, and finally is the in vitro culture of the endosperm tissue. So the first is the natural selection or occurrence of the triploids naturally. Though the frequency of the natural triploids is very low, but they do originate spontaneously in nature. In nature, triploid may originate from the fertilization of unreduced gametes with the normal gametes. Their frequency is usually low as their seeds are inviable and hence these plants cannot propagate through the sexual propagation. So these do not have progenies and therefore even when they are spontaneously appear or they spontaneously created in the nature, they disappear with the completion of their life cycle. Typically, these plants can be identified morphologically because of some specific characters, like they are usually faster growing as compared to their diploid progenitors. Their organs are usually larger, like they bear the large fruits or large flowers, and also they do not make the seeds, so seedlessness is another very important feature for the identification of triploid. So here is a megaspore or a microspore mother cell of a species which has two sets of chromosomes, that is diploid. Now these will undergo meiosis that will result in the formation of male gamete and female gamete, that is pollen and egg respectively. And these gametes have half the number of chromosomes as that of the somatic cells, and it is represented by N. These gametes fuse to form the zygote, which develop into the sporophyte plant. So that's how the normal life cycle of the sexually propagating plants proceeds. However, under certain circumstances, due to the meiotic errors, reductional division may not occur and therefore it results in the formation of unreduced gametes. That means gamete formed as a result of non-reductional division would have the chromosome number same as that of its parent's chromosome number. So the ploidy level of these gametes would be same as that of the ploidy level of the body cells of the parent. So this gamete has two sets of chromosome. When these unreduced gametes, that is the gamete with two sets of chromosomes, they will fertilize with the normal gamete, it will result into a triploid zygote that will develop into a triploid plant. Now the question is that what are these circumstances that lead to the formation of these unreduced gametes? Usually the environmental stress conditions such as wounding, frost, cold, drought stress, nutrient shortage, etc. may stimulate the production of the unreduced gametes. In fact, stress has been a driving force behind the expansion of the polyploid plants among the angiosperms among the land plants. Another method of triploidy production is the fusion of gametes from two species of different ploidy level, like the fertilization of the gametes formed by a diploid species with the gametes formed by a tetraploid species. These will also result into the triploidy. Like here it is an species B, that is a tetraploid. It means that it has 4 copies of chromosomes. So each chromosome has 4 copies, or each gene has 4 alleles. During the meiosis, it will produce gametes that would have 2 sets of chromosomes. So here the ploidy of the normal gametes formed by a tetraploid species is similar to the ploidy of the unreduced gametes formed by the diploid species. So they have 2 sets of chromosomes, their ploidy is 2x. Now if there is a cross-pollination between the species A and B, fertilization of these gametes with 1 set of chromosome and 2 sets of chromosome would result into a triploid zygote. Triploidy usually result in the increase in the size of the body cells or the somatic cells, specifically the guard cells in the leaf, and also it increases the total number of the chloroplasts in the cells. These features strengthen the photosynthesis, and therefore many triploids are more vigorous, resulting in a higher accumulation of the biomass, and that further result in the increased crop yield. Some of the plants for which the natural triploid plants have been identified include Populus species, like the Populus tremula or Populus alba are the triploid. They are spontaneously created triploids. Some of the species of Quercus are also triploid. Quercus is the oak tree genus. And then the annual Miscanthus species, which belong, it is actually a grass species which belong to the family Poaceae, and it is being explored for the production of a biofuel. So some of the triploids have been identified for the Miscanthus species also. So the natural triploid or the spontaneous creation of the triploids do occur in the nature, however their frequency is very low. Next is the artificial hybridization. Now the sexual hybridization of a diploid and tetraploid is the most common method for the production of the triploid plants through the human intervention. We discussed on the last slide that it can occur spontaneously or naturally also. However, the natural occurrence of spontaneous tetraploidy is rare, as therefore frequency of natural triploid is also very low. Further, because these triploids do not propagate sexually, so most of the times spontaneously originated or natural triploids, they are lost with the completion of their cycle, life cycle, if they are not protected or if they are not multiplied by alternative methods. So basically, triploidy can be achieved by fertilization of either egg from a diploid and a pollen from the tetraploid species, or vice versa, that is the egg is from a tetraploid species and a pollen from a diploid species. Tetraploidy can be induced artificially by chromosome doubling, which is achieved either by the application of the spindle inhibitors during the cell division, or it can also be achieved by the fusion of the protoplasts. As we discussed earlier too in the in vitro androgenesis, that there are several chemicals that can be used for the spindle inhibition, causing the doubling of the chromosomes. However, colchicine is one of the most commonly used inhibitor and has a very high success rate for the chromosomal doubling. Other frequently used spindle inhibitors are orazoline and trifluralin. Even after having the diploid and tetraploid species, the successful production of triploid plants depend upon several factors like pollen viability, compatibility between the diploid and tetraploid parents, or the frequency of unreduced gametes if we are using the diploid parents, and the stress for the production of the unreduced gametes. Therefore, stress can be applied during the reproductive phase for the increasing the frequency of the unreduced gametes. In addition to the effect of the environment on the pollen viability, genotype has a huge impact on pollen germination and growth of the pollen tube too. Usually the female parent has a huge impact on the success rate of the hybridization. During these artificial hybridizations, three different types of seeds have been obtained, that is undeveloped seeds, then the developed seeds but they are very small in size, and third category is developed seeds of the normal size. It is the developed seeds of the normal size which are most useful, and other types, that is the undeveloped seeds or the developed seeds with the smaller size, they do not make or germinate into adult plants. Overall, triploidy has achieved through artificial hybridization by developing the tetraploid cultivars. Now, artificial induction of triploidy can be induced by polyspermy also. Polyspermy is the fertilization of more than one male gamete with the egg cell, and it can result in the higher ploidy too. Usually after the fertilization of male gamete with the egg cell, fertilization of the second male gamete is prohibited by the genetic regulatory control, and this phenomena of blocking the second fertilization or fertilization of the second sperm with the fertilized egg is called as the polyspermy block. However, the fertilization of second nuclei can be induced artificially, and hence triploids can be regenerated or can be created by artificially inducing the fertilization of two male gametes with the egg cell. So in this case, gametes from the male and female gametophyte can be isolated, and then the fusions are performed in vitro. So here is an egg cell and a male nuclei. The fusion can be induced by using some of the fusion inducing chemicals or through the electrofusion. So it results into the entry of the male nuclei into the egg cell. Now within a few minutes of this first fusion, second male nuclei can be fused with the fertilized egg, again through the electrofusion or using some other available tools for supporting this fusion. This results in the fusion of the second male nuclei, and now the egg cell has three nuclei, that is one of its own nucleus and two male nuclei that has been resulted or that has been fused with this egg cell. Now further development may occur either of the two pathways. One option is that two male nuclei fuse to each other in the egg cell, and then this nuclei with the two set of chromosomes, that is, it has a two exploidy, it fertilizes with the egg cell, thereby generating the triploid zygote. Alternatively, the one male nuclei can first fuse with the egg cell or egg nucleus, thereby generating a diploid zygote. However, it is followed by the fusion of the second male nuclei immediately, which result in the zygote with the three chromosome sets, that is, it is triploid, its ploidy is 3x, and this further develop into a triploid plant. Though very little work has been done in this direction, that is the artificial induction of the triploidy by artificial polyspermy, but it has a huge potential to contribute for the development of the triploid. Next is the endosperm culture. Now let's have a quick look on the double fertilization in angiosperms. Double fertilization is the typical feature of the angiospermic flowers or the sexual reproduction in angiosperms. So this is a typical structure of angiospermic flowers having different organs like sepals, petals, male reproductive organs and female reproductive organs. During the pollination, pollen is received by stigma here. Once the pollen lands on the stigma, it germinates and the pollen tube grows from the stigma towards the ovary through the style. Pollen tube carrying two male nuclei enter the female gametophyte through the synergids in the ovule and releases these two male gametes in the embryo sac. So here is a pollen tube carrying two male nuclei is entering into the embryo sac through one of the synergids. Finally, it releases the two nuclei in the embryo sac. One of the nuclei fuses with the egg cell and form the zygote, which will later form embryo and germinate into the complete plants. And the second male nuclei fuse with the secondary nucleus in the central cell, which is itself a fusion product of the two nuclei of the female gametophyte. So therefore it already has a two sets of chromosome, and the fusion of the second male nuclei with the secondary nucleus results into a triploid cell which develop into an endosperm. This endosperm is a nutritive tissue and provide the nutrition to the growing or germinating embryo. Therefore, during the fertilization in angiosperms, one male gamete fertilizes the egg cell to form the zygote, and the second male gamete fuses with the two nuclei in the central cell of the embryo sac that forms the endosperm. And this phenomena of fusion of two nuclei with the two different cells of the embryo sac is called as a double fertilization. So this triploid endosperm tissue is naturally formed in the angiosperm plants, and it can be used to regenerate the triploid plants through in vitro cultures. So basically, like other plant cells, endosperm cells are totipotent and has potential for the unlimited growth. Therefore, the endosperm tissue can be used for the in vitro culturing. As it is a triploid in nature, the in vitro regenerated plants from this tissue would be triploid too. Though endosperm is the nutritive tissue for the developing embryo or germinating embryo. Its amount varies among different plants, from very little in the legumes or the cucurbits to a huge amount in case of the cereals. So the seeds where the endosperm is little or reduced, the nutritive function is usually taken up by the cotyledons. Now the triploid plants can be regenerated through the in vitro culture of endosperm tissue. And in fact, the endosperm tissue has been successfully cultured for several plant species like rice, citrus, neem, eucalyptus, etc. So there are several factors like the impact of the genotype or the developmental stage of the endosperm that can impact the in vitro regeneration, and I will discuss these factors in a few minutes. And finally is the regeneration of the triploid plantlets. Like the other tissue culture regenerated plants, it may occur through the two mode of differentiation, that is either direct regeneration, that is the tissue do not undergo, so the endosperm tissue do not undergo dedifferentiation to form the callus. So the endosperm cells directly divide and they form the embryos and germinate into triploid plants. Second pathway is the indirect regeneration. So that is the first explant tissue dedifferentiate. So the endosperm tissue dedifferentiate to form the callus. And then morphogenetic responses or the shooting responses are induced from the callus using various hormonal combinations. Usually, if the starting material is mature endosperm, it dedifferentiate and proliferate to form the callus. That is, it follows indirect mode of regeneration. And from immature endosperms, both type of regeneration pathways have been observed. Endosperm culture, one of the most direct and most efficient method of triploid production. Though the endosperm culture appears to be a straightforward approach for the production of triploids, but there are several challenges for in vitro culturing of the endosperm tissue. First is the development of mixoploid chimeras that appear frequently during the endosperm culture. Usually when we cross-hybridize species of two different ploidies, progeny plants may have mixture of ploidy plants. Similar type of the chimeric plants originate in the endosperm cultures too. The endosperm tissue in general may have several ploidy levels and that may exist at the same time in the different cells, and therefore when this tissue with the different ploidy levels or having the cells of different ploidy are used for the in vitro cultures, it may result in the chimeric plants or plants may have the different ploidy. Even the development of the aneuploid plants have also been observed frequently in the endosperm cultures. Further, the cells with the different ploidy level in the endosperm may respond differently to the concentration of the hormones and type of the plant hormones. So therefore, ploidy of the regenerated plant may vary with the type of the hormone used or the concentration or the combination of these plant hormones used to regenerate the plantlets in the in vitro culture from the endosperm. It has been observed that the induction of the callus proliferation from the endosperm tissue is comparatively easier and has been achieved for several plant species. However, it is the induction of the morphogenetic responses or the shoot regeneration from the callus that has a very low success rate. In fact, for several plants only callusing has been achieved, and the percentage of the plants where the plantlets were regenerated successfully is very, very low. In several cases, complex media have been used for the in vitro cultures of the endosperm. Now, the composition of these complex media is not clearly known, and the quality or the composition of this media may vary from one lot to another lot. And therefore, usually the reproducibility of the endosperm culture or the in vitro culture using the complex media is not uniform. Next is the factors that has direct impact on the endosperm culture. There are several factors that directly impact the responses of endosperm tissue on the in vitro culture media. These can be grouped into the three categories. These are impact of the genotype, developmental stage of the endosperm, and finally the composition of the culture media. Now, first is the genotype. Now, the endosperm response on the in vitro culture is genotype dependent. That means the potential of the endosperm cells for in vitro responses varies significantly among various plant species. Efforts have been made to culture the endosperm from several species, but the success stories are available for only a few plants like rice, citrus, eucalyptus, neem, etc. For many of them, though the culture have been established, but the regeneration of the triploid plantlets could not be achieved. Hence, the regeneration from the endosperm tissue is technically challenging, and the optimization of the endosperm culture could be a tedious process. Overall, it appears that the callus induction is comparatively easier and can be achieved for most of the plant species that were tested for the endosperm culture. It is the regeneration or the induction of morphogenetic responses which is critical and need to be optimized. Next is the impact of developmental stage of the tissue or the sampling time of the endosperm tissue for in vitro cultures. Now, the age or the developmental stage of the endosperm can impact the endosperm culture considerably and is critical. However, there is no consensus if any particular development stage is critical in all the plants. It has been observed that while in some species immature endosperm have been used successfully for culturing, like in maize it is about 8 to 10 days after pollination when the endosperm is more responsive. However, in other plants, it's the mature endosperm that gives a better response, like the Jatropha or the Ricinus. So, if you compare the immature embryo versus the mature embryo, it is the mature embryo which produces a better result with the triploid production in these plants. Further, the initial association of the germinating embryo with the endosperm is critical for inducing the cell division in endosperm. Usually, mature endosperms do not proliferate to form callus if the embryo is removed before initiating the callus. However, once the cell division starts in the endosperm on the artificial media, embryo can be removed and it does not affect the further development of the endosperm culture. And so it has been suggested that the germinating embryos can contribute some factors that may be essential for inducing the cell division in endosperm tissues. However, once the division is initiated, these factors may not be required for further development of the dividing endosperm. So the presence of endosperm or presence of the embryo for initiating the cell division or callusing from the endosperm tissue is essential. Next is the impact of the culture media on the endosperm culture. Now, MS basal media is most commonly used for the endosperm culture. However, it needs to be supplemented with other nutrients. As earlier also I talked about that the complex media have been successfully used as a supplements with the MS media and the cultured endosperm successfully. Some of the commonly used such supplements or the complex media includes potato extract or the tomato juice, corn juice or the yeast extract, etc. Use of the plant hormones like auxins or the cytokines is essential for the endosperm culture. For the induction of the callus, usually use of 2,4-D is more successful in most of the plant species that have been tested. Among cytokinins, benzyl aminopurine, that is BAP, is more successful. However, other hormones like naphthalinic acid, indole-3 acetic acid, or zeatin or kinetin have also been used successfully for the endosperm culture or the culturing triploid plants from the endosperm. Supplementation of the MS media with the organic source of the nitrogen is essential and has a considerable impact on the successful cultures. And usually casein hydrolysate is used for this purpose. For the regeneration of the shootlets, the concentration and the ratio of the plant hormone is the key factor. However, it varies among different plants and need to be optimized in the species specific manner. Overall, though there are several challenges or the obstacles that decreases the probability of regenerating the triploid plants from the endosperm culture, however, it is still a common method of producing triploids. Because of the time required is less as compared to the other method, and also the success rates appears to be better. Next is the application of the triploids. Now, triploids are rare in nature as their seeds are not viable and hence they lack the progenies. However, because of their several beneficial features, considerable progress has been made to understand the biology of the triploids, and they are being used in the agriculture, horticulture, or the forestry, or some other industries. First of all, triploids are sterile, and hence they are desirable for the production of seedless fruits. Several triploid fruits are popular in the food industry like the seedless grapes or banana, apples or seedless watermelons. And the percentage of the seedless fruit in the market is continuously increasing. Here is an example of the production of seedless watermelon. Now the watermelon are diploid plants and therefore in order to develop the triploid watermelon, we can use either the endosperm culture or hybridization between the diploid or tetraploids. Here, hybridization between the two species of different ploidy have been used for generating the triploid watermelon, and it's more successful in this case. So the chromosomal doubling, so here is a watermelon, basically it is a diploid watermelon, and in the diploid watermelon, chromosomal doubling is induced by the application of the spindle inhibitors, and it results in the production of a tetraploid watermelon. Now these tetraploid watermelons are crossed with the diploid plants. So here the pollen from the diploid plants are used to fertilize with the egg from a tetraploid plant. So basically female plant is used here is a tetraploid and the diploids are the male plants. So the pollen from the diploid plant is used in these crosses. So the female contribute the 2 sets of the chromosome in the egg cell, and the male contribute 1 set of the chromosome in the male gametes. When these gametes fertilize, they produce the hybrids that are that have 3x or 3 sets of chromosomes. So they have a triploid ploidy. When these plants are grown, these plants bear seedless fruits. These triploid plants are self-infertile, and therefore a standard diploid cultivar is also used along with these triploid plants for the purpose of pollination. Diploid cultivar used for pollination or for this purpose are selected in such a way that either these are early maturing as compared to the triploid, or they should have some specific morphological features like different ring color or the different color pattern of the fruits. This will help, or these morphological features of the diploid plants help to identify the diploid plants or separate the diploid plants from the triploids. Next is the prolonged flowering period. Though the triploids are sterile, their flowers are generally larger and more colorful as compared to the diploids, and are of huge interest commercially in the horticulture market. The larger size is possibly because the energy devoted for the seed development is invested in the flower development and its maintenance. These flowers have longer shelf life, and hence these are better performers. Therefore, cleaning of the faded and dead flowers, which is essential for maintaining the plant's appearance and performance, is either not required at all, or even if it is required at a minimal level. So overall efforts required are less for the maintenance of these triploid plants. Overall, it results in the higher returns. Then is the photosynthetic efficiency. Now, triploids usually have a larger somatic cells and the guard cells. Further, they have higher number of chloroplasts in their body cells as compared to the diploid counterparts. Therefore, it leads to a better photosynthetic efficiency, and these all characters results in a relatively more vigorous nature that result into the accumulation of higher biomass and better crop yields. Next is the neutralizing invasiveness. Now, the introduction or the movement of the plant from one region to the other region is very common these days because of the commercial interests. However, when these are introduced to the new environment, the plant may proliferate vigorously, and it can actually impact the local biodiversity or the ecosystem, causing severe environmental or economical losses. Invasive plants usually produce large amount of seeds that is responsible for their quick or rapid proliferation. Therefore, for the plants of the commercial importance that have invasive nature, seedless triploid varieties can neutralize the invasive nature of these plants and can be very helpful for the introduction of these plants into the new environment or in the new ecosystems. Further, triploids can have a better nutritive value too. Like the protein content in the leaves of the triploid mulberry is about 4% higher than the diploid mulberry. Therefore, these leaves have a better digestibility for the silkworms. In fact, when the silkworms were fed with the triploid leaves only, the silkworm grew rapidly and their life cycle was also reduced by few days, and the overall productivity was also increased significantly. Triploids are useful in several ways in the research too. Like these can be used to study the genome evolution, that is the imbalance in the gene doses and its consequences. And these can be used to study the genome rearrangements too. So in this way, these Triploids are helpful to study the genetic regulation or how the different genomes they interact to each other. Overall, triploids are very important commercially in the agriculture and the horticulture industries. During the last 2 decades, significant progress has been made with respect to optimizing the methods to make the triploids. Endosperm culture is the easiest and rapid method for triploid production. However, development and optimization of the new tools would be required to extend this method to the plants that are usually do not respond to the in vitro cultures. So in summary, we discussed about the triploids today, various methods that can be used to create the triploid plants. Specifically, we discussed the endosperm culture in little more detail, and then the various factors that affect the endosperm culture, and finally the various applications of the triploids in agriculture, horticulture, or forestry. Thank you.